Welcome to the deep dive. Today, we're driving into the world of Streptococcus pneumonia.
A fascinating one.
It is. And, for this deep dive, we're using Principles and Practice of Clinical Bacteriology, 2nd edition.
Ah, Gillespie and Hockey.
Exactly. It's a great resource, really packed with information. So we're going to try to pull out the most important stuff for you, especially if you're a medical microbiologist.
Perfect. Because, S. pneumonia, it's a bacteria we see all the time in the lab, right?
All the time. So let's get started. First things first. How would you describe S. pneumonia? For someone who's maybe never seen it under a microscope.
Well, it's tiny.
Tiny.
Yeah, and it's this gram-positive coccus. But it's really interesting. It's got this lanceolate shape.
Lanceolate.
Like almost a football shape.
Oh, I see.
Yeah, it's pretty distinctive.
So we've got the microscopic view.
Mm-hmm.
But what about when we see it, growing on a plate?
Ah, yeah, the colonies. You'll see these, how do I describe them? They're kind of dome-shaped at first. But then, get this, they collapse in the center.
They do. They cave in. Why is that?
It's because of autolysis. The bacteria kind of self-destruct. And when that happens, they release this, it's a key virulence factor called pneumolysin.
Pneumolysin. So that's what's doing the damage.
Yeah, it breaks down red blood cells. That's why you see that alpha hemolysis around the colony.
The greenish halo, right?
Exactly. But it does a lot more than just that.
Okay, so it's not just about what we see on the plate. It's a big player in how the bacteria causes disease, too.
Oh, absolutely.
Gotcha. So we've got the shape, the colonies, the hemolysis. What else helps us ID S. pneumonia in the lab?
Well, you've got your classic tests, right? Optogen sensitivity.
Right.
It's inhibited by optogen. And then there's bile solubility. And, of course, it's catalase negative.
Okay, so those are the key things to remember. But here's the big question, right? How does something that looks so simple under the microscope cause so many different and serious infections?
It's a good question. Because S. pneumonia, it's got this whole arsenal of virulence factors.
An arsenal.
Yeah. It's how it sticks to our cells, invades tissues, hides from our immune system. The whole works.
Wow. Sneaky little buggers.
They are.
So let's break this down. What's the first step? How does it even get a foothold?
It all starts with adhesion. S. pneumonia has to stick to our cells before it can cause any real trouble.
Makes sense.
And it does this with these, they're like grappling hooks on its surface. They latch onto cells in the respiratory tract, the middle ear, you name it.
Grappling hooks, huh? Interesting analogy.
Yeah.
So are all the strains equally sticky?
That's where things get really interesting. S. pneumonia actually comes in two forms, transparent and opaque.
Transparent and opaque.
Yep. And these forms, they have different capsules and surface proteins, which changes how well they stick to things.
So it's like they have different outfits for different jobs.
Yeah. Exactly. The transparent ones, they have thinner capsules, they're sneaky, they can slip past our immune system, and they're really good at sticking to cells, especially those epithelial cells.
So they're the ones causing the invasive infections, right? Pneumonia and meningitis.
Yeah, you got it.
What about the opaque ones? What are they up to?
They have thicker capsules, more armor, you could say, but they're not as good at sticking to those epithelial cells. So you're more likely to find them just hanging out, colonizing, not necessarily causing those big infections.
But trade-offs, huh? Stealth versus resilience.
Exactly. It's how S. pneumonia survives, adapts. That's what makes it so tough to deal with. So
you mentioned those grappling hooks, those surface proteins that help it stick. Any specific ones we should know about?
Oh, yeah. There are a couple of key players. One is called choline-binding protein A, CBPA for short.
CBPA. Got it. And what does that one do?
It's a multitasker. It helps S. pneumonia stick to cells, invade tissues, even messes with our immune system.
Wow, a real triple threat. How does it manage all that?
Well, it binds to this molecule called choline.
Choline.
Yeah, and choline, it's all over the surface of our cells. So CBPA, it can basically latch onto any cell it wants.
Like a master key.
Pretty much. And once it's attached, it can trigger all sorts of things.
Like what?
It can make the cells take up the bacteria. For one, it can disrupt how the cells work. It's pretty amazing, really.
So CBPA is definitely one to watch out for. Any other surface proteins we should be aware of?
Yeah, there's another important one called pneumococcal surface adhesin A.
Okay, that's a mouthful. Pneumococcal surface adhesin A.
Yeah, but we usually just call it PSA, and it's really important for the bacteria to bind to cells in the nasopharynx.
The nasopharynx, so that's where it starts.
Yeah, that's where it first sets up shop.
And then from there, it can spread.
Exactly. But what's interesting about PSA, it also acts as a transporter.
A transporter. What is a transporter?
Manganese.
Manganese. What's so special about manganese?
It's actually crucial for S. pneumonia to survive. It needs it for all sorts of processes, and it helps protect the bacteria from damage.
So it's like it's fueling itself up, getting ready for battle.
You could say that.
So we've got CBPA and PSA both helping S. pneumonia stick around. What's next? How does it go from just hanging out to actually invading tissues?
That's where things get even more interesting. Well, S. pneumonia, it has these enzymes. They're like molecular scissors.
Molecular scissors?
Yeah, they can cut through things, break down barriers. Like one of them is called hyaluronidase.
Hyaluronidase?
Yeah. Have you heard of hyaluronic acid?
I have.
It's what holds our tissues together, part of that extracellular matrix.
Right. Yeah.
So hyaluronidase, it breaks that down, basically makes little gaps for the bacteria to squeeze through.
So sneaky.
I know, right? And then there's neuraminidase, another one of those enzymes.
Neuraminidase. Okay, and what does that one do?
This one's interesting. It chops off these sialic acid molecules from our cells.
Sialic acid.
It's this sugar molecule. It's on the surface of a lot of our cells.
Okay, so the bacteria just snips it off. What's the point of that?
Well, it's kind of like clearing the way. You see, when it removes that sialic acid, it exposes receptors that S. pneumonia can then use to attach even better.
Ah, I get it. It's like cutting down the trees to see the forest, or in this case, the receptors.
Exactly. Pretty clever, huh?
Very clever.
Yeah.
But our immune system, it's got to be fighting back, right? We can't let these bacteria just waltz in and take over.
Oh, of course. Our immune system's always working. But S. pneumonia, it's got ways to fight back, too.
Oh, I bet.
Remember PSA?
The manganese transporter.
Yeah, that one. Well, it turns out it also helps S. pneumonia evade the immune system.
Really? A transporter and an immune evader. That's impressive.
It is. It messes with our complement system.
The complement system, remind me what that is again.
It's part of our innate immunity, one of the first lines of defense.
Okay, right.
So PSA, it binds to this protein called C3. And that blocks the whole complement cascade from getting started.
So it's like disarming one of our weapons before we can even use it.
Exactly.
Wow. Anything else PSA does on the immune evasion front?
Oh, yeah. It stops another complement protein, C3b, from sticking to the bacteria.
What's that do?
Well, when C3b sticks to bacteria, it makes them a target for our phagocytes.
Phagocytes being?
The cells that gobble up the bad guys.
Right. So PSA is protecting S. pneumonia from getting eaten.
Exactly. So it's getting the manganese it needs, and it's hiding from our immune system, all thanks to PSA.
Talk about a multitasker. It's amazing how much this one little protein can do.
It is, isn't it? But there's one more virulence factor we can't forget about.
Which one's that?
Pneumolysin, the one we talked about earlier.
The one that causes the alpha hemolysis.
Yeah, exactly. It's released when the bacteria lies, and it can cause all sorts of trouble.
Like what?
Well, for one thing, it's a pore-forming toxin.
A core-forming toxin.
It means it can punch holes in our cell membranes.
Oh, yikes.
Yeah, it can actually kill cells that way. Not just red blood cells, either.
So it's targeting other cells, too, not just causing the hemolysis we see on the plate.
No, it can damage all sorts of cells. Lung cells, cells in blood vessels, even immune cells.
That's scary. Anything else?
It also activates the complement system.
I thought you said PIA was blocking the complement system.
It does, but pneumolysin, it activates it in a different way.
What do you mean?
Well, instead of helping clear the infection, it actually makes the inflammation worse.
Oh, no.
Yeah.
So it's like the bacteria's turning our own defenses against us.
Yep, that's one way to think about it. And that's not all. Pneumolysin can also mess with our cilia.
Cilia. Those little hairs in our respiratory tract.
Exactly.
What does it do to them?
It slows them down. They can't sweep away mucus and debris as well.
Ah, so that's why people with pneumonia have such a bad cough. The bacteria are basically crippling our cleaning system.
Exactly. And that makes it even harder to clear the infection. So pneumolysin, it's a real troublemaker.
It sounds like it. S. pneumonia really has a whole arsenal of weapons, doesn't it?
It does. It's a well-equipped little bugger.
It's amazing how much damage something so small can cause. It's fascinating, but also a little scary.
I agree. But it's important to understand how it works. That's how we can figure out the best ways to fight it.
That's true.
Yeah.
So we've talked about the bacteria itself, its virulence factors, but all of this, it's happening inside a person.
Yeah.
It's causing real problems.
Of course. And that's where the clinical side of things comes in.
Exactly. So let's shift gears a bit. Let's talk about what S. pneumonia actually looks like in patients. You know, the clinical features of these infections.
Sure. I mean, you've got your common ones, right? Pneumonia, meningitis, otitis media, sinusitis.
Right. Let's start with pneumonia. What does that look like in a patient?
Well, classic case. You're going to see fever, chills, and this pleuritic chest pain, like a sharp pain, gets worse when they take a deep breath.
Oh, I see.
And they'll be coughing like a lot.
productive cough.
Yeah, at first the sputum might be whitish or yellowish, but as it progresses, it can turn rusty or even blood-tinged.
Okay, and all that makes sense, right? Fever, chills, that's the inflammation.
Yeah.
The pleuritic chest pain, that's because the lining around the lungs is inflamed. And the cough, that's from the bacteria messing with the airway.
Exactly, you're putting it all together.
Well, I'm trying. What about when you examine the patient? What do you find?
You can often hear crackles when you listen to their lungs.
Crackles.
Yeah, it's this kind of bubbling, rattling sound. It means there's fluid in the air.
Fluid from the inflammation.
Exactly.
Okay, so you're hearing these crackles. What else?
Well, you can also feel for consolidation.
Consolidation.
Yeah, it's when a part of the lung fills up with fluid and cells becomes solid. Sounds dull when you tap on it.
Oh, interesting. So you're putting together the symptoms, what you hear, what you feel, but how do you confirm it's S. pneumonia?
You got to look for the bacteria itself.
Right. Back to the lab.
Yep. You can get a sputum sample, look for those gram-positive diplococci under the microscope, culture it on blood agar, see those draftsman colonies.
The ones that cave in?
The very same. And then do those other tests, optochin, biosolubility.
Make sure it's S. pneumonia. Got it. So pneumonia, that's one big one. You mentioned meningitis, too. That's a lot more serious, right?
Oh, definitely. With pneumonia, it's the lungs. With meningitis, it's the brain and spinal cord.
The meninges, right?
Yep, the membranes that cover the brain and spinal cord. When those get inflamed, you see all sorts of neurological symptoms.
Like what?
Well, often starts with a really bad headache,
Yeah.
stiff neck, and they can't stand bright lights.
Meningism, right? The classic triad.
Exactly. But it can get a lot worse than that. Seizures, confusion, coma. It can be really scary.
And how does it even get to the brain? I mean,
Right.
It starts in the respiratory tract, right?
It does. And it's not always clear how it gets to the meninges. Sometimes it travels through the blood.
Through the blood.
Yeah, it crosses the blood-brain barrier.
The blood-brain barrier. That's
Yeah.
supposed to protect the brain from things like bacteria, right?
It is, but S. pneumonia, it can find ways to sneak through, especially those transparent variants.
The sneaky ones.
Yep, they're good at crossing that barrier, and once they're in, they can trigger this huge inflammatory response.
So it's not just the meninges that are inflamed.
Nope, it can spread to the brain itself, cause encephalitis, that's swelling of the brain, and that's when you see those really serious complications.
It's amazing how something so small can cause so much trouble.
I know, it's pretty humbling.
So how do you even diagnose meningitis? Is it like pneumonia with the cultures and stuff?
Well, you definitely look for the bacteria, but the gold standard for diagnosing meningitis is a lumbar puncture.
A lumbar puncture, that's a spinal tap, right?
Yep. You get a sample of cerebrospinal fluid, the fluid that surrounds the brain and spinal cord.
Okay. And what are you looking for in that fluid?
Lots of things. You'll see a higher white blood cell count, mostly neutrophils.
Neutrophils are.
They're a type of white blood cell. They're the first responders to infection.
Got it.
The protein level in the fluid will be high, and the glucose level will be low.
Low glucose. Why is that?
The bacteria are using it up. They need sugar to survive.
Ah, clever little buggers. So you're looking at the cells, the protein, the glucose. Can you actually see S. pneumonia in the CSF?
Sometimes you can do a gram stain, look for those diplococci under the microscope, and you can try to culture it too.
So it's kind of like a puzzle. You're putting together the symptoms, the exam findings, and the lab results to get the whole picture.
Exactly.
Okay, so we've covered pneumonia, meningitis. What about those other two you mentioned, otitis media and sinusitis?
Ah, those are super common, especially in kids.
Otitis media, that's a middle ear infection, right?
Yeah. The kid will have ear pain, fever, maybe some drainage from the ear. They'll be fussy, pulling at their ear.
Oh, poor things.
And sinusitis, that's a sinus infection. So facial pain, pressure, stuffy nose, thick green or yellow mucus.
I've had that. Not fun.
Nope, not fun at all. And both of these, they often happen after a cold or the flu.
The viral infection, right?
Exactly. The virus kind of sets the stage, makes it easier for S. pneumonia to move in.
So it's like a 1-2 punch.
That's a good way to put it.
So how do you diagnose these?
Otitis media, you can usually tell just by looking in the ear. The eardrum will be red and bulging.
Oh, I see. And sinusitis.
That one's trickier. You have to go by symptoms, maybe do a CT scan to see if the sinuses are inflamed.
So it's a bit more detective work.
It can be.
Okay, so we've talked about what these infections look like in patients. Let's talk about who gets them. The epidemiology, right?
Right. So who's most vulnerable? It's the very young and the very old.
The extremes of age. Makes sense. The immune system isn't fully developed in babies and toddlers, and it weakens as we age.
Exactly. But there are other risk factors too, like any condition that weakens the immune system. HIV, diabetes, chronic lung disease, heart disease, you name it.
So it's not just about age. It's about how strong your immune system is.
Exactly. And then there are anatomical things too, like if your spleen isn't working properly or if your cilia aren't sweeping things away as well as they should.
Okay, so age, immune system, anatomy, anything else? What about the environment?
Oh, yeah. Environment definitely matters. If you're in close contact with lots of people, like in a daycare or a nursing home, that increases your risk.
So it's more likely to spread in those settings.
Exactly. And things like cigarette smoke, that can damage your lungs and make you more susceptible to.
So it's a mix of things, right? The bug, the host, the environment, they all play a role. What about genetics? Does that come into play at all?
It does. There's this gene called the Ficaria R131 allele.
Wow, that's a mouthful. Ficaria R131 allele.
Yeah, it's a bit of a tongue twister. But basically, if you have a certain variation of this gene, you're more likely to get really sick with pneumococcal pneumonia.
Interesting. So your genes can influence how you respond to the infection.
Exactly. And that's why personalized medicine is so important. Maybe someday we can test people for this gene and know who's at higher risk.
That would be amazing. Speaking of prevention, let's talk vaccines. There are vaccines for S. pneumonia, right?
There are two main types, actually. You've got the 23-valent polysaccharide vaccine.
23-valent polysaccharide vaccine.
Yeah, it's called PPV23 for short.
PPV23, got it. And what does that one do?
It targets a bunch of different serotypes of S. pneumonia, but it only triggers a T-cell independent immune response.
T-cell independent, what does that mean?
It means it doesn't give you really strong, long-lasting immunity.
Oh, so it's not the best.
It's okay for healthy adults, but it's not great for little kids or older folks. Their immune systems just don't respond as well to this type of vaccine.
Okay, so what's the other type?
It's called the 13-valent pneumococcal conjugate vaccine.
13-valent pneumococcal conjugate vaccine. That's even longer.
I know, right? We call it PCV13 for short.
PCV13. Much easier.
Right. So PCV13, it targets fewer serotypes, but it triggers a T-cell dependent immune response.
Which is better.
Much better. You get a stronger, longer-lasting immune response.
So that's the one you'd give to kids.
Exactly. It helps them build up good immunity early on when they're most vulnerable.
It's amazing how these vaccines work, like tricking the immune system into being prepared.
It is. It's one of the most powerful tools we have to prevent disease.
So we've prevented as much as we can. But what if someone still gets sick? What do we do then?
Then it's time for treatment.
And what does that look like?
Well, the mainstay of treatment is antibiotics.
Antibiotics, okay. But I remember you saying something about antibiotic resistance earlier.
Oh, yeah, that's a big problem. We're seeing more and more resistance to penicillin, macrolides, even some multi-drug resistant strains.
That's worrying. So what can we do?
Well, for susceptible strains, penicillin is still the go-to. Usually they give it intravenously, especially for serious infections like pneumonia or meningitis.
Okay, penicillin if it works. What if it doesn't?
Yeah.
What if it's a resistant strain?
Then we have to use something else. Third-generation cephalosporins like ceftriaxone or cefotaxime, those are often good alternatives.
Cephalosporins, okay. And what about macrolides?
We try to reserve those for less serious cases or for people who are allergic to penicillin.
But you said there's resistance to those too, right?
Yeah, unfortunately. The bacteria have figured out ways to block them or pump them out of the cell. It's a constant battle.
It's like they're always one step ahead.
Sometimes it feels that way.
So it's really important to use antibiotics wisely, right? Not overuse them.
Yeah.
Make sure we're using the right one for the job.
Absolutely. Antimicrobial stewardship, it's crucial. We have to protect the antibiotics we have while we're working on developing new ones.
So it's a team effort, right? Doctors, pharmacists, patients, we all have to work together.
Exactly.
Okay, so we've talked about prevention, we've talked about treatment. What about the management of these infections? Is there anything special we need to do?
Well, the most important thing is to diagnose and treat quickly. The sooner you start antibiotics, the better the outcome, especially for serious infections.
Makes sense. Time is of the essence.
It is. And then there's supportive care, making sure the patient is comfortable, managing their fever and pain, keeping them hydrated.
Treat the whole patient, not just the infection.
Exactly.
Are there any specific things you do for the different types of infection?
Sure. For pneumonia, sometimes people need oxygen therapy if their blood oxygen level gets too low.
Okay.
And in really severe cases, they might need a ventilator to help them breathe.
Wow. So it can get pretty serious.
It can. With meningitis, you really have to stay on top of things, make sure the inflammation doesn't get out of control.
And how do you do that?
Well, sometimes we use corticosteroids like dexamethasone to reduce the swelling. And you have to monitor them really closely, watch for any signs of trouble.
So it's a delicate balance, treating the infection but also protecting the brain.
Exactly. For otitis media and sinusitis, it's mostly about relieving the symptoms.
Yeah.
Pain relievers, saline sprays, things like that.
So a more conservative approach.
Usually, yeah.
Yeah.
Unless it's a really bad case or it keeps coming back, then you might need antibiotics.
Okay, so it really depends on the individual case.
It does.
Well, this has been incredibly informative. I feel like I've learned so much about S. pneumonia.
Me too. It's always fascinating to delve into the details of these little buggers.
They're definitely more complex than they seem at first glance.
Oh yeah. They're masters of adaptation, always finding new ways to survive.
It's a reminder that we can never let our guard down. We always have to be learning, researching, developing new strategies to fight back.
Absolutely.
Okay, before we wrap things up, let's put your knowledge to the test. I've got 5 exam questions for you.
Bring it on.
All right. Question number 1. What's the key thing that makes S. pneumonia look different from other alpha hemolytic strep under the microscope?
Oh, that's easy. It's the shape, that lanceolate shape, like a little football.
Exactly. 1 point for you. Okay, question number 2. A patient comes in with fever, chills, that stabbing chest pain when they breathe, and they're coughing up rust-colored sputum. The chest x-ray shows a whole lobe of the lung is consolidated. What's the most likely culprit?
Sounds like a classic case of pneumococcal pneumonia.
2 for 2. You're on a roll. Okay, next one. Which of S. pneumonia's many weapons is responsible for that alpha hemolysis we see on blood agar?
That would be pneumolysin.
3 for 3. You're S. pneumonia expert.
Yeah.
All right, question 4. We've got 2 main vaccines for this bug. Can you name them and tell me the key difference between them?
Sure. We've got PPV-23, which covers a lot of serotypes, but doesn't give you the strongest immunity. And then there's PCV-13, which covers fewer serotypes, but gives you a much better immune response.
Perfect. That's 4 out of 4. Last one. You're suspecting meningitis, so you do a lumbar puncture. What are you looking for in that spinal fluid that would tell you it's S. pneumonia?
Well, you'd see more white blood cells than usual, especially neutrophils. The protein level would be high and the glucose level would be low. And if you're lucky, you might even see the bacteria themselves under the microscope.
5 for 5. You've aced the exam.
Thanks. It's been fun.
It has. I've learned so much today. I hope our listeners have too.
Me too. It's amazing how much there is to know about this one little bacteria.
And it's constantly evolving, which makes it even more challenging.
That's for sure. It's a reminder that we always have to be on our toes, ready to adapt and find new ways to stay ahead of the game.
Well said. Well, that brings us to the end of our deep dive into the world of Streptococcus pneumoniae. We hope you've enjoyed the journey and learned something new along the way.
Thanks for having me.
It's been a pleasure. And to our listeners, thank you for joining us. Until next time, stay curious and keep exploring the amazing world of medical microbiology.
It really is amazing, isn't it, how S. pneumonia has managed to stay so relevant, causing so much trouble for so long.
I know, it's like a tiny little bacteria with a huge impact.
Exactly. Thinking back on everything we've talked about, what do you find most interesting about it, like from a microbiologist's point of view?
Honestly, it's the fact that we're still learning new things about it, like those opacity variants. That was a game changer.
Yeah,
right?
Totally. It's like we thought we knew S. pneumonia, and then boom, it throws a curveball. Really shows you can't get complacent in this field.
Exactly. There's always something new to discover.
Which is what makes it so exciting. So what are researchers focused on now? What are the big mysteries left to solve when it comes to S. pneumonia?
Well, vaccines are a big one, right?
Yeah.
PCV-13 has been great, especially for kids.
But S. pneumonia is smart. It's evolving.
Right. New serotypes are popping up.
Yeah.
And some are even becoming resistant to the vaccine.
It's like a constant arms race. We develop a weapon, they find a way around it.
Exactly. So what are researchers doing about that?
Well, they're working on new types of vaccines, protein-based vaccines, for example.
Protein-based. How are those different?
They target parts of the bacteria that don't change as much. So they could be more effective against a wider range of serotypes, even new ones that pop up.
So kind of a more universal vaccine. That would be amazing.
It would. And then there's the microbiome. That's another hot topic in S. pneumonia research.
The microbiome, what's that got to do with it?
It turns out the other bacteria living in our noses and throats can influence whether S. pneumonia can colonize or not.
Really? So it's not just about S. pneumonia itself, it's about the whole community of bacteria.
Exactly. And if we can understand those interactions, maybe we can find ways to manipulate the microbiome, make it harder for S. pneumonia to take hold.
That's fascinating, like fighting bacteria with bacteria.
Exactly. It's all about understanding the ecosystem.
Well, I have to say, this deep dive has really opened my eyes to the world of S. pneumonia. It's so much more complex than I ever imagined.
I agree. It's a truly remarkable organism, even if it does cause us a lot of trouble.
It does. But knowing how it works, how it causes disease, that's how we can fight back, develop better treatments, better prevention strategies.
Absolutely. It's all about knowledge and using that knowledge to improve human health.
Well said. Well, that brings us to the end of our deep dive into the world of Streptococcus pneumoniae. We hope you've enjoyed the journey as much as we have.
Thanks for having me. It's been a great discussion.
The pleasure was all mine. And to our listeners, thanks for joining us. Until next time, stay curious and keep exploring the fascinating world of medical microbiology.